Welcome, everybody. Welcome, welcome. Thank you for joining us. Yes, please drop where you're from in the chat. Welcome to the workshop. I know we're across many time zones, so thank you for joining us wherever you are. We're going to go ahead and get started. So, people who are in some later time zones can go ahead and get to sleep. All right. And we'll go ahead and keep admitting people. Um, all right. Okay. Let's go ahead and get started. All right, everyone, welcome. My name is Shaquisha. I am the Career Pathways Manager for Hack the Hood. Welcome to the Melanin Science and Hack the Hood Bioinformatics Workshop, where tech meets life sciences. Today, we have a wonderful, wonderful workshop and a wonderful guest speaker today. And I hope you all learn a lot. Thank you again for being here, and let's jump in. So, before we get into the actual workshop, I want to start with a little bit about who Hack the Hood is. So, we want to start with who these organizations are who are co-hosting this event for y'all. So, Hack the Hood is a tech education nonprofit, and we provide Black, Latinx, API, and Indigenous youth with tech skill building programs and career navigation support that's grounded in social justice and support economic mobility. So that is who we are, what we do, and our values. And I'm going to pass it to Carmen Banks to talk a little bit about Melanin Science and who they are and what they do.
Hi, everyone. Welcome. Thanks so much for coming. And for folks who are on different time zones where it's late or super early, I really appreciate you stopping by to join us for this for this bioinformatics workshop. My name is Carmen. I'm the CEO and founder of Melanin Science. And we are an organization that's committed to building the future of science by creating pathways for folks like yourself into biotech and biopharma careers. And we do that through different educational modules like this bioinformatics workshop, industry tours here in the Bay Area. I'm in San Francisco, and also by cultivating your professional and soft skills for biotech and biopharma jobs.
Awesome. Thank you, Carmen. And next, we are going to have Aneesa, our wonderful speaker, introduce herself and talk about who she is.
Thank you so much. It's a really generous intro. My name is Aneesa. I'm based in Brooklyn, New York. I'm from Brooklyn with Caribbean heritage, and now I work in software for the life sciences, but I'm a sort of a bioinformatics scientist by training. People are often surprised to hear that I dropped out of my PhD. So if any of you in the comments are considering PhD programs or are currently in PhD programs and you're kind of maybe conflicted, to Carmen's point, we all dropped our info in the chat. So feel free to hit me up outside of this workshop. But yeah, I also recently launched my own company. It's not here because it's like a month old. It's called ImpaktBio, which is here but backwards for some reason. And we're a training company. We do stuff like this to just enable learners and enterprise businesses to work with biology data at scale. So really happy to be here and happy to hopefully impart some knowledge.
Amazing. Thank you so much. And I'm going to stop sharing right now and have Anissa take over so we can start our demo.
Yes, let's see. Hmm. I hate when the Zoom tab is in the way. One second. Is this showing as full screen for folks or is it tiny? Yeah?
Nope, you're
Okay.
good.
Okay, great. So I'm gonna actually start with a few slides because I feel like based on the backgrounds in the audience, and thank you all so much for completing our very thorough questionnaire. I think we maybe need to orient ourselves a little bit before we dive into the code. So this should take maybe the first half hour or so. And feel free to stop me with any questions at this point, because once we get to the code, I'm probably going to ask folks to save the questions till the end, just so we can get through all of the material. Why is that not working? Oh, okay. So we're going to talk first about the biology, because quite a few folks here actually come from adjacent specialties. So people are in chemistry, people are in like energy, mechanical engineering, that sort of thing. And then we're going to talk a little bit about compute and computation and how that is important for answering questions in biology. So first things first, this is an exercise in bioinformatics. And bioinformatics is a pretty new field. I'd say it emerged within the past 10 or so years. And it's a very interdisciplinary field. It draws from computer science, from math, especially statistics, and of course, biology for domain knowledge. And I think actually folks tend to use computational biology and bioinformatics interchangeably, but there's a slight nuance. Um, Sorry, folks are still coming in and they're popping up. There's a slight nuance in the outcomes, pretty much. So computational biology is sort of the overarching domain that uses statistical modeling to do a lot of system simulation, whereas bioinformatics, we're also using statistical modeling, but specifically to analyze large-scale data sets. That's like a very niche application of bioinformatics, which is what we're going to do in the code. Why is that not working? Okay, there we go. So if you are new to biology and you don't listen to anything else that I say for the next hour and a half, just know the central dogma because this underlies all of biology. It was postulated by Francis Crick in the 1950s, and it basically says that this is how information flows in a cell, pretty much. DNA is transcribed into mRNA, which is then translated into complex polypeptides, which form proteins. And I think this is a really simplistic rendering of this sort of process, but for our purposes, I think this is fine. And the flow of information from DNA to mRNA to polypeptides, we can consider that gene expression, so to speak. So you might be wondering, well, why should I care about gene expression? Why should we quantify gene expression? And that's because transcriptomic profiles can tell us a lot about particular cell states, which can help inform a lot of different things. One being helping us understand cellular identity and function. So different cell types, cell populations are going to have unique transcriptomic profiles. What makes a muscle cell doesn't necessarily make a skin cell or a blood cell or what have you. Quantifying gene expression can also help us identify disease mechanisms. So you might imagine that certain genes in someone who has cancer are going to be very different than the genes that are turned on in someone who's healthy or someone who has Alzheimer's. Of course, quantifying gene expression can help us understand drugs and develop drugs and test drugs. If you imagine that, okay, well, my transcriptomic profile as someone who has not been treated with, I don't know, aspirin, just some over-the-counter drug, is going to be different from that of someone who is being treated with aspirin for a long-term chronic issue, for example. By now, hopefully you're starting to see like a pattern in that we're doing the same thing in all of these instances, quantifying gene expression. We're just sort of switching the circumstance. So in number one, we're looking at gene expression across different kinds of cells. Two, we're looking at gene expression across different disease states. Three, gene expression between healthy and drug treated. Exact same for four. We're just looking at gene expression across, let's say, embryonic development. So like E1 versus E16 and looking at which regulatory networks are changing within that time frame. And as you can imagine, all of these help us make progress towards personalized medicine, which has been pretty big, let's say, in the past 10 years, and just developing solutions and treatments that are personalized to the individual person rather than to a group of folks. Okay, so excuse me, we talked about what is gene expression. We talked about why should we care about quantifying gene expression. So how do you actually do that? Well, that is a laboratory process. What we're going to do today is a dry lab exercise. It's fully computational, but quantifying gene expression actually happens in a laboratory, a wet lab at the bench by laboratory scientists. And so this was sort of the one of the earlier methods of quantifying gene expression, and it's called bulk RNA sequencing. So essentially what happens is you have a piece of tissue, you kind of like crush it up in a beaker, you extract the RNA, and then you're able to quantify gene expression at the cellular level or cell population level. So the advantages of this is that there's no need for any probes. You'll see why that's important. It yields a really comprehensive transcriptome profile, and it's highly sensitive and specific. So this was popular through the advent of like next-gen sequencing in the early 2000s. And this was pretty standard commonplace for quantifying gene expression in academic settings, in industrial settings. This was the standard. So fast forward a little bit, the technology evolved, methods became more sophisticated, and quite literally, biology just became increasingly complex. And so we needed to now quantify gene expression at the single cell level, which is what we're gonna be working with single cell data in the code. And the difference here is that you get a piece of tissue, again, you crush it up in a beaker, but the output is measured at the single cell level. So instead of getting an output where you say, okay, these are the genes that are expressed in muscle cells versus blood cells. Single cell RNA-seq allows you to say, okay, this is the transcriptomic profile of a single cell, an individual cell in your body. So as you might imagine, this is extremely high resolution. It produces detailed cellular profiling, and it helps us to identify rare cell populations that bulk RNA-seq might miss because you're just crushing entire tissue and getting a population level readout, as opposed to this extremely granular single cell level readout. So this is now the standard for academia and industry as it relates to quantifying gene expression. Okay, how does it actually work? I'm probably going to not mention a lot of the detail here, but this is how you actually isolate RNA and quantify gene expression. Again, this happens in a wet lab, so this is upstream from what we're going to be doing today. What happens, in short, is that you encapsulate single cells in oil, like literal oil, and then you add these bar-coded beads to your suspension. And these bar-coded beads are important because they each have unique molecular identifiers. And we're going to see why that's important because after this step, you lyse the cell, which is just a fancy word for you break it open. And then the RNA that's attached to the bead hybridizes, or just that's a fancy word for it binds to the RNA in the cell. And now you've got this suspension of lysed single cells, so just RNA floating around, that's attached to the beads that you added in. And the beads are important because that's how you identify the individual single cells. Like you need to have a way to quantify, to trace back the individual cell. These are all unique identifiers so that you can say, okay, this is cell one, cell two, cell three, through cell 1 million. And the output of this is DNA, specifically cDNA that you've just reverse transcribed your RNA. But all you need to know is that after this RNA extraction, you've got DNA. And that's important because that's going to serve as the input for this next step, which is the actual single cell sequencing. And this can get pretty convoluted, but essentially you take the output here, your cDNA, you attach adapters to either end, you attach it to this flow cell, you amplify it through PCR, which basically just means that you make several copies of the DNA. And then there's this fancy laser on the inside that measures the intensity of the DNA at particular points, and then it assigns a letter based on the intensity. So if it says, okay, the intensity at this point is greater than 80%, that's going to be an A. And it's going to do that for the four nucleotide bases. So that's how you get DNA sequences, A, T, C, G, T, T, A, C, what have you. And these are stored in FASTQ files. Okay, so we're almost done. I'm telling you all of this because all this needs to happen before we get to what we're going to do today, which is just analyzing the count matrix. So now you've got a bunch of DNA or reads, short reads, DNA sequences from your cell sample that you crushed up, you broke open the cells, and you hybridized the RNA to. So now you need to make sense of that because all you're looking at is just hundreds of lines of letters at this point that's stored in a particular file. So to make that information make sense, you need to align those reads to a reference genome. And a reference genome is actually exactly what it sounds like. It's an entire genome where we know every single gene in that genome. And so we can take the reads that we've produced and map those to that genome. Imagine you've got like a hat of words and you've got a dictionary where you know every single word and its meaning, its associated meaning is in that dictionary. So if you pull any word out of your hat, you can map it to the word that's in the dictionary because that's the source of ground truth. It's the exact same thing here. The reference genome is your dictionary, and this set of reads are all the new words that you've got in your hat. And this process is called alignment. And this is actually also a computational exercise. We're just not going to do that today. So at this point, we've walked through a lot of the upstream steps in a single cell analysis pipeline. So we've talked about the reads, those long nucleotide sequences that we got from after our RNA isolation and all those steps. You generate a count matrix from this step, actually. I overlooked that. Let me go back a little bit. As you are aligning your reads to the reference genome, what you do is accumulate a set of counts because these reads encode genes, right? So every read that you align counts one time. If you align the same gene to the reference genome, it's going to count twice. Another gene counts three times. So what you end up with is a ton of genes with their associated counts in a file. So that is the count matrix that's generated here. It's a matrix of cells by genes and their associated counts. We're going to start here today. We already have a count matrix. So what we're going to do is filter that matrix um, to sort of get rid of low quality cells. And then we're going to do a bunch of like pre-processing or integration steps. We're ultimately going to cluster the data and identify some markers. In reality, this is a pretty cyclical process. We're not going to loop through this today, but in real life, it's never a linear, simple process. And then once you've got some markers, you can do a ton of different stuff. You can do trajectory analysis, you can do a differential gene expression analysis, which we are going to do, or any sort of custom analysis based on the biological question that you might want to answer. Okay, that's it for biology. Let's talk briefly about compute and why it's important. So compute in biology has become important probably within the past 10 years as well, actually, and we'll see why in upcoming slides. But now we're sort of partnering with computers. Maybe 5, 10 years ago, I would say as a wet lab scientist, maybe you didn't need to know how to analyze your single cell data. But now I actually think it's not optional to at least have a little bit of computational knowledge. It enables us to do a lot of things. One, because now we've just got too much data. We quite literally do not know what to do with it, how to store it, how to handle it. Because of advances in technology, the complexity of biological systems can actually sort of be evaluated now, and at least in ways that we weren't able to do 5, 10 years ago. And because of that, we need systems that are efficient, systems that can get us insights quickly. I think the current bench to bedside pipeline for drug development is north of 10 years now, which is a very, very long time, and it's extremely expensive. So we need innovations in compute to sort of decrease that window. And of course, for predictive power, we're not going to talk about drug development today, but doing things like predicting 3D protein structures to determine where drugs might bind to particular targets is extremely, extremely important and lucrative. So just to give folks an idea of how far we've come, this is what the genomics landscape has looked like. We started in the late 1900s with Sanger sequencing, excuse me, and it was extremely slow and laborious, and it produced like hundreds of base pairs of sequence per experiment. So as you can imagine, we really didn't care about compute power and just like distributed computing and efficient storage or any of that at that point in time. Fast forward though to the late 1900s to the early 2000s, a lot of you might remember when we sequenced the first human genome, and this required significant algorithmic innovation because this produced 3 billion base pairs compared to the hundreds only that we were able to produce with Sanger sequencing. So at this point is sort of when the compute wave started, when people realized that, okay, we've generated all this data, how do we store it? How do we access it so we can actually do things with it? And then fast forward a little bit more, almost to present day, but not quite, to the next gen sequencing era. The single cell pipeline that I just showed you from Illumina, that's when Illumina got pretty big. The only difference here is that we're still producing billions of short reads, but we are now producing billions of short reads in parallel. So before, this was a sequential operation. We had to sequence one human, then we could sequence the other, then we could sequence the other. But now we can sequence multiple in parallel, which brings us to sort of the age of big data genomics, which is where we're currently at. And a lot of the efforts in modern day genomics are focused on efficiently storing and accessing the data. And it sounds easy and it sounds trivial, but we're talking about like tera and petabytes of data that we want to do stuff with. And so now there's huge demand. A lot of biotech startups, including mine, are focusing on efficient storage and retrieval, specifically of like scientific unstructured data. Okay, so we're almost done. That brings me to the data that we'll be using today. This is, some of you might be familiar with the Chan Zuckerberg Initiative, but in recent years, they've curated what is the largest single cell atlas in the world. I think this number is a little bit off, 60 million. It's now sitting at 74 million single cells and about 65,000 genes. So remember I told you it's a count matrix. So imagine 74 million rows, individual single cells, and then 65,000 columns or genes. And this is a really, really rich data set, as you might imagine. There are multiple demographics represented here. There are multiple disease states represented here. Um, yeah, it's just honestly, it still kind of blows my mind that this exists. Shameless plug, my company, TileDB, is the storage backend for the CZI census data. And importantly, this is interoperable, meaning that you can analyze this data in Python, you can analyze it in R, you can do whatever you want. We're going to do Python today, though. And if you want to play with this link is not doing what I needed it to do. But if you want to play with this data, definitely just Google CZI cell by gene census and it'll pop up. They've got like a whole interactive tool if you don't know how to code and that sort of thing. Okay, so this is the workflow for the next hour. We're going to take a small slice of the census data because we're certainly not using 74 million cells or 60,000 genes. My machine can't handle that, so I don't imagine that you guys can. We're going to take a small subset of that data. We are going to save it or store it in an and data object. If you code in R, you should know Seurat objects. And data objects are the exact same. They're just the Python equivalent. And then we're going to do some stuff to make the data good for identifying highly variable genes. Then we are going to cluster the data by disease. You can cluster it by whatever you want, actually. But I'm going to cluster it by disease. And then towards the end, we're going to shift a little bit from thinking about individual genes to thinking about signaling pathways and taking a more systematic approach. So that's it. But I think, sorry to the folks in the chat. You guys were covered this whole time. I know I said stop me with questions. So I'm just going to go back into the chat now and see.
I can field the questions for you, Aneesa. So, first question was, this was kind of during the process of you talking about the RNA-seq on the biology end, but they asked if it was a similar process to taking a DNA test.
If single cell RNA sequencing is similar to taking a DNA test, yeah, they do need to sequence your DNA in the same way. All of that stuff is abstracted from the average, person in a doctor's office, though. Unless you're doing genetic testing, then I think they actually do give you your FASTQ files, which is that those files are just like lines and lines of nucleotides. But yeah, that's the first step, the single cell sequencing.
The next question is, which tool do you use for alignment to your reference genome?
Oh, star. Sorry. So yes, this wasn't just a random word. Star is a command line tool. So yeah, we could have also done that today, but that takes more than an hour for sure. There are a few star BCF tools. That's all I can remember for right now. But star is the standard for single cell RNA-seq alignment.
And the last question right now, Mark asked, their lab has a database of mRNA sequence of rats, and they are saddled with the responsibility of deriving useful information from the sequence for human benefit. How can they go about that? And I think you'll probably go through that process shortly.
Yeah, so the question is you've got rat single cell sequences, and they want to derive biological meaning from that. Is that it, Carmen?
Database of mRNA sequence of rats,
Mhm.
and yeah, finding useful information from the sequence for human benefit. Mark, if you're available and come off mic, you can also ask your question.
Yeah, it's exactly what I asked. I work in a lab where they have sequenced the mRNA of rats. And then one of my responsibilities was for me to compare the sequences that we have derived from rats with that of human, and then derive something meaningful from the data, and then suggest some possible research that we can do and follow up to that. So I've been, confused about the whole thing, and thank God for this webinar.
Yeah, of course. So, there's a couple things that you could do. In general, I would say if you have rat sequences, then you first need to align those sequences to a rat reference genome. Here. So there are reference genomes for every model organism. So you can align to a rat reference genome and then look at if your genomes are from rats under special circumstances, like if you have genomes from rats who were just chilling as rats, and then you have genomes from rats who were maybe treated with a drug, or a lot of rat research is like cortisol stuff. So maybe you did like a behavioral study on that other group of rats and you've got those genomes. Then exactly what we're gonna do today, you can do for your rat experiment, looking at things like differentially expressed genes, clustering by disease if that's important to you, or maybe cell type, gene set enrichment analysis, which is a sort of like a signaling pathway analysis if you're trying to map human behaviors to rat behaviors. Like if rats are just your model organism for understanding a certain human behavior, then a gene set enrichment analysis could work. There are so many things. Yeah, hopefully that gives you a good start.
And if folks have questions, we'll just put them in the chat. I'll field them. And then when Aneesa is done with the demonstration with the data, then we'll go back and answer those questions.
Okay, great. I'm going to stop sharing and reshare. One second, I'm just moving you guys out of the way. Hold on. Oh, okay, great. Okay. Carmen, you guys can see this.
Yep, you're good.
Great. So I'm gonna probably explain things more than I typically would just because of the backgrounds in the audience. So before we even start on this code, I'm gonna orient us. What you're looking at is my integrated development environment, or my IDE. This is Visual Studio Code, or VSC. This is typically what I use to develop code. PyCharm is another IDE that folks might be familiar with. It's a little older. Sometimes if I'm developing code across multiple GitHub repos or just across like several complex projects, then I'll use Cursor. Cursor is an AI assisted code editor, and it looks a lot like this actually. There's just a chat element. So this is VSC, and I've already set up my virtual environment for us today, and I can probably send this out after actually, with including the virtual environment, so you guys can try this on your own. But this is a notebook that I put together detailing an end-to-end single cell analysis pipeline. This is certainly not an exhaustive pipeline. These are just maybe the most common methods that folks use in single cell analysis. And we're going to do a few different things here. So we're going to, like I mentioned, we're going to grab just a small subset of that 74 million cell data set. Then we're going to do some quality control or some QC. And the point of that is to sort of filter out low quality cells because biology is not perfect. We're also going to normalize and log transform the data. And this is just to prep the data for downstream analysis, like isolating highly variable genes. Then we're going to cluster our data specifically by disease, but like I mentioned, you can do whatever you want when you get your hands on this notebook. And then we're going to close with some pathway analysis. All right. So what I'm going to do here is just import my dependencies. For the folks who are new to programming, this is how you start no matter what you're doing. At least if you're programming in Python, you install your libraries or your dependencies. And these are just pieces of software that hold different functionality. So here I'm installing packages that help me handle data, that help me manipulate data frames. I'm installing packages that help with data visualization, so creating plots, which we're going to do. And here I'm installing a ton of bioinformatics specific packages. So these are packages that are going to help us bring the data in. These are packages that are going to help us manipulate the data for the gene set enrichment analysis. And this one in particular, TileDB Soma, is one of my company's APIs. It's going to help us access the data from storage. So that's done. Now we're gonna slice off a chunk of the first 5,000 cells and only 10,000 genes from that CZI cell by gene data set. So here we're just specifying we only want the first 5,000 rows and the first 10,000 columns. Remember, this data set is like millions of cells big. So this is a very, 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 very small fraction of that. And here, I just want to make sure folks understand this because the end data object is what we're going to be working with. So we just pulled in a slice of that data from the cell by gene census, and we stored that data into what is called an end data object. And you see here it says these are the dimensions, observations, or rows, and variables, columns. We've got 5,000 rows, well, 5,001 because I think it took the indices, but we've got 5,000 rows, 5,000 single cells, and we've got 10,000 genes. What we also have in this and data object are metadata associated with our samples. So all of these names right here are columns that exist in the data. It's just more information about the cells themselves, the samples. And then all of this here is metadata specifically associated with our genes. So more information about the genes. And I'm going to show you what that looks like. So this is what OBS looks like, the OBS layer of our and data object. And this head function is only printing the first 5 rows. So we've got some different cell types in here, endothelial cells, malignant cells, and some other stuff. We've got breast cancer. These cells were sampled from someone with breast cancer, at least the first 5 of them. And there was something else I wanted to point out. Where is it? I can't remember, but it's okay. It's not gonna run this one. And this is just if we want to take a look at the disease states that are represented in the data. Because remember up here, this head function, we're only printing the first 5 rows. So just because we see breast cancer in this column doesn't mean we've only got breast cancer samples in the data, which is why we want to use this unique function on the disease column in our OBS data frame to print the unique values in that column. So this is actually what we've got in our data. In 5,000 rows and 10,000 genes, we've got 3 disease states represented. We've got samples from folks with breast cancer, from healthy individuals, and from folks who have Alzheimer's disease. And this is just an output of the total number of disease states that are represented in the data. So we've only got 3. because remember, we only sliced this small subset of the data. But in the million cell atlas, there are 138 disease states represented, some of which are listed here. Okay, so now I just want to show you what var looks like. Remember that this is metadata associated with our genes. So this is information about our genes. You can see here, this feature name column represents our actual gene names, but we've also got other information. So we know that whatever this is a long non-coding RNA. Whatever this is a protein coding RNA. And this sort of metadata exists online. If you've ever heard someone say that they're annotating their single cell data, this is exactly what it means. Based on these unique ensemble IDs, you can append this metadata onto your data if it doesn't already exist. For us, this already exists because this is a pretty popular data set. Okay, so now let's switch gears a little bit. So we've ingested our data. We know what it looks like. Now we want to start pre-processing the data for actual analysis. And one of those things is to, just as a sanity check almost, to make sure that there are no duplicate genes in our data, because that is going to interfere with quite literally all of our downstream analysis. That's one of the assumptions that we make in biology in single cell analysis, is that when we are doing any sort of operation on a count matrix from single cell RNA sequencing, we assume that no gene is duplicated. Every single gene that's in the data is unique. There's only one instance of that gene. So thankfully, we don't have any duplicates in our data. Otherwise, we would remove them. Now we're going to move on specifically to QC. Remember I said that biology is not perfect. I think if I could impress anything upon you is that as a computational biologist, bioinformatician, whatever you want to call it, we can't just not acknowledge the biology and the imperfection of biology. Because remember, I showed you all those upstream steps happen in a lab before we get to this point. And the technology in the lab is not perfect. Of course, human error exists. And so the point of quality control is to ensure that we're acknowledging the imperfection of biology. We are trying to make our data set clean, so to speak, get rid of low quality cells so that any insights that we generate are reliable and reproducible, which are some of the core tenets of just scientific inquiry. So one way to do that, to do QC, is to identify mitochondrial genes in your data. And we want to do that because mitochondrial, or the activity of mitochondrial genes in a cell, can be an indicator of a lot of things. A cell with high mitochondrial activity might be stressed. It might be a doublet, meaning something could have gone wrong upstream, and instead of isolating a single cell, now we've got two that emerged for some reason. And conversely, a cell with aberrantly low mitochondrial activity could be dying. So in either extreme, we don't want those cells in our data. So first, what we want to do is check for any mitochondrial genes in the data. Thankfully, in this data, we also don't have any, but they're typically annotated with MT. So if we were to look here in our var data frame, in our list of genes, our column of genes, we would see MT 1, 2, 3, etc. And that would denote a mitochondrial gene. If we had any mitochondrial genes, then based on the percentage of mitochondrial genes activated in the data, we would drop certain cells. But we don't have to worry about that with this data set. So because we don't have any mitochondrial genes, we can actually use some of the QC metrics that we already have in our and data observations. So here, sorry, you guys are in the way again. So here we've got a few sort of aggregate statistics. We've got a raw sum, which is a count number. We've got an N and Z value, which essentially is like a number of non-zero values per cell. This is just an indicator of the number of unique gene features in a cell. And we've got some other stuff. We've got means, we've got variances, and so forth. Those are less important. But those are the metrics we're going to use to establish some evidence-based cutoffs for filtering our data. So remember the raw sum that we just saw, that's going to be the total number of counts per cell. So remember when I showed you the splice-aware star alignment, aligning your DNA to the reference genome and then generating that count matrix? Each gene in a cell has a particular number of counts. And so that cell has a total number of counts, a count of all genes, instances of those RNA transcripts in that cell. So that's what this raw sum number is. And then this N and Z, number of non-zero genes per cell, you can think of that as like a feature number, like a unique feature number. So the count is the total number of instances of all genes in the cell, and the N and Z, or the frequency, are like those unique instances of all genes in a cell. If it doesn't make sense now, maybe it'll make more sense once we do the plots. So this is just a sanity check for me. I'm just renaming my columns from raw sum to N count RNA, and from N and Z to N feature RNA, because these two are just more standard. Like I prefer to see them. That's really all it is. And here we're actually going to do the plot. So we grab the values in each of these two columns, and then we do a violin plot, and we overlay a strip plot onto those violins. And here's what that looks like. So what you see, if you've never seen a violin plot before, the violins are these colored shapes, sort of like in the background, and the strip plot are these dots in the foreground that are overlaid onto the violins. Violin plots help give you a picture of the distribution of your data. So this like fat part is where the majority of your data points are concentrated, and same on the right side here. And then they taper off into a tail, which basically just tells you that, okay, you don't really have many data points in this region. And then I overlaid the strip plot because I felt like it would be easier to actually see the data. So each one of these little black dots is a single cell in both violins. And I think it's good to plot your data because I don't know about the folks in the audience, but I personally am a visual person. So actually seeing the distribution of my data helps me decide, okay, well, I want only cells from here to 2000, or from here 100 to 5000 or something, which is exactly what we're going to do. So actually, before I go to that, before I run that next cell, this is unfortunately not an interactive workshop, which is what I typically do. But at this point, I would ask you, well, how would you interpret these violin plots? If I'm telling you that this is where the bulk of your data points are concentrated around the fat parts, and then they taper off into sort of nothingness, then how would you, if you were to set cutoffs, by set cutoffs, I mean if you were to remove cells from your data based on what these violins look like, how might you set your cutoffs? And I would remind you that the N feature RNA number is like the total instances of unique genes in a single cell. And this count number is sort of less important, but it's the total number of counts of all genes in a particular cell. So that question was rhetoric, um, because it's not interactive. So I'm just going to answer it and tell you what I would do. So I would set my cutoff for n feet, the number of features at 100, my lower bound at 100, and my upper bound, max genes here, I would set at 4,000. So I basically want in my data the cells starting from the very bottom here till around here. And this is a really subjective process because some of you might have said, oh, well, I would stop it at 5,000 or maybe even at 6,000. This is where biology, it's kind of eye of the beholder sort of thing. But that's where I would set my cutoff. I consider everything above 4,000 to be an outlier or just not that impressive. And then I've set my N count RNA value to, I just want everything above or equal to 100. So I basically just want everything here.
Aneesa, can you just give us a little bit more context on why those cutoffs are needed?
Yeah, for sure. So let me go back. So this establishing the cutoffs are necessary for doing quality control on your data set. We started our quality control here by looking for mitochondrial genes, because remember that mitochondrial genes are an indicator of cellular health. Whether cells are stressed, they'll have a lot of mitochondrial activity, or cells are dying, they'll have aberrantly low mitochondrial activity. So that was the first exercise in doing quality control. This part, plotting the total number of features and counts in individual single cells, helps us filter out low quality cells. Because if I'm looking at this is the distribution of my entire data set of the features, the unique features in my data set, then reliably I can say, anything above maybe here probably would be considered an outlier and just not informative in my data or just not significant enough for me to include in my data. The purpose of doing this is to ensure that we've got a data set that is sort of just robust. Like we want high quality cells. We want as many of them, sorry, as we can get. And knowing that we're willing to sacrifice, a couple hundred cells or so to just ensure that we've got a really robust data set. I think this makes more sense with extremely large data sets. Um, I probably should have I probably should have prefaced this by saying we will not find anything interesting here today because our data set, this is super small. We're only working with 5,000 cells and 10,000 genes. When you get into like the 100,000 cell range or the million cell range, then this QC step becomes essential because the single cell sequencing, the process that I showed you guys is also not perfect. Sometimes cells aren't isolated independently. So sometimes you've isolated two cells at once instead of a single cell. And so you can't get any sort of reliable transcriptomic profile from that because now we've got two cells. So it's not single cell in the same way that it needs to be. So that's why we're doing this. We just want to get rid of low quality cells because we want a robust data set. Hopefully that helps. Okay. So now, oh, my bad. So after my filtering, I'm down to 4,961 cells and the same number of genes because we haven't filtered genes. So after my filtering, I got rid of roughly 40 low quality cells. So we're still pretty good. That's a good ratio. Now that our quality control is done and our data set is robust, so to speak, we can do a lot of math and stats to prep it for analysis. One of those things is going to be normalizing and transforming the data so that we can identify highly variable genes. And then we're going to have to scale the data for PCA, and I'll tell you what PCA is and why it's important. And then we are going to cluster and identify our cell types with a UMAP plot. So I'm going to run this. What we're doing here is first normalizing our data. And this is important because we want to start on sort of like a level playing field. There are a few characteristics of single cell data that make it not great for analysis. One of those things is the fact that single cell data is actually inherently sparse. So a lot of the values are actually zero values. The counts are actually zeros. And another characteristic of single cell data that makes it not great to do anything with is that you've got cells with extremely high gene expression values and cells with extremely low gene expression values. And that sort of skew is not good for downstream analysis. So the normalizing here is that we're normalizing each cell to 10,000 counts per cell while still preserving sort of the intercell variability. So we're still preserving the proportion of gene expression of all the genes in each cell across two different cells, but we are normalizing each cell to 10,000 counts. So imagine if you've got like 3 cups of water. 1 cup is full, 1 cup is like almost empty, and the last cup has water to the middle. We are making all our cups equal. We're gonna make them all have water at the midline mark. That's what we're doing for each of these cells. And then we're gonna transform the data. We're gonna do a log transformation. And what that does is it compresses the ranges. So I just mentioned that, some cells have extremely high gene expression values and some cells have extremely low gene expression values. We want to compress the total range so that the values between cells are more comparable. I did tell you guys that bioinformatics is a lot of stats too. So we've done that. This is a sanity check for me. I'm just making it very explicit that I want my var to use this column for my features because this column has our gene names. You don't have to do this. That's just my OCD. Okay. So now we have data that's been filtered. We did QC. We normalized the values to each cell is going to have 10,000 total counts. We log transformed the values to compress ranges. Now we can calculate highly variable genes in our cells using this package from ScanPy. And what this does is, sort of simply put, it calculates a mean across a gene expression mean across all the cells, and then it also calculates a variance, and it evaluates how far the variance of each gene is from its calculated mean. So long story short is that it gives you, based on this, it gives you a list of highly variable genes, the genes that are changing significantly in your data. And we're asking for only the top 1,000 highly variable genes because we're only working with 5,000 cells. So that's done. Now we're going to plot it. And this is what that looks like. So we've got our genes on this y-axis and then a percentage of total counts on the x-axis. Just visually, I mean, this is pretty self-explanatory, but the further along on the x-axis, the more highly variable the gene, which is why this malati, or is that malat one? This why this gene is first. Okay. So actually, before I get to this, identifying highly variable genes is important for if you're doing an experiment and you want to determine the effect of maybe a drug or some sort of compound, maybe a vitamin, um, then this is helpful because you can identify highly variable genes in your healthy cohort versus which genes are highly variable in your treatment cohort. All right. So bringing it back to characteristics of single cell data that make it challenging to just do anything with. Another one of those characteristics is the fact that single cell data is extremely highly dimensional. What that means is we are talking about millions of stuff on the y-axis, one dimension, and millions of stuff on the x-axis or a second dimension. And the dimensionality makes it hard to do downstream analyses because, I mean, as humans, we can only interpret three dimensions for all my organic chemistry people on the call. And so before we do anything with the data, we need to reduce the dimensionality of the data set not only for our interpretation, but for the algorithm's interpretation. Because you can think of dimensionality, high dimensionality, as kind of like a needle in a haystack sort of situation. So you are trying to isolate the needle or whatever biological signal that you are interested in, but you're just surrounded by hay. You're just surrounded by noise. You're surrounded by all these empty, non-informative data points because there's just so many of them. So that's why we're going to do PCA, which is a dimensionality reduction method before we do our clustering. Before we do the PCA though, we need to first scale the data, and scaling is different from the normalization and the logarithmic transformation we did up here. Scaling, where is it? Scaling tries to bend the data into a normal distribution. So we want all our values to centralize around a mean of 0. And we also want to cap outliers at a particular standard deviation. So here I made this note that this argument caps extreme z-scores to mitigate outliers. So past a score of 10, absolute 10, so positive 10, negative 10, the algorithm caps the data. And this is important because PCA assumes normally distributed data, which definitely is not what single cell data is. So that's why we have to first scale it. And then we're going to actually perform the PCA, which is just one line of code. And then we're going to visualize the PCs or the principal components. So PCA, the output of PCA are these PCs or these principal components. And you can think of them as like bags of variance. So each PC is responsible for a particular amount of variance in the data or importance in the data. PC1 is responsible for the most variance in the data, and the importance or the significance of the variance decreases as you go to the right, as far as ranking goes. So the first few PCs are typically the most important, and then we don't really care about from like PC 12 to 31. But this is called an elbow plot. And at this point is when I would be interactive again and ask you, oh, why do you think it's called an elbow plot? Um, Rhetoric. I'm just going to tell you it's an elbow plot because somebody decided that this looks like an elbow, like that this curve looks like an elbow. There's a point in this curve where it just bends like an elbow. It's a whole thing. Anyway, this is an elbow plot, and the way that we interpret this is also subjective, like many things in biology. But essentially, you want to almost insert a bend mentally in this plot to give you an indication of which PCs are most important. So for me, if I'm looking at this, I would probably, if I'm being generous, I would probably cut my PCs here between PC 6 and PC 7. If I'm being a little more stringent, I would probably cut them here between 4 and 5, or even 3 and 4. But essentially, this on its own is not informative. This is just to give us a sense of how much of the variance is contained within the first 1 to 4, 1 to 5, 1 to 6 PCs. We're going to keep all of these principal components up to 30. We're going to save them in our AND data in a different layer, so not OBS or var, but this OBS M layer, which is like metadata for OBS. And we're going to use the PCs to compute a neighborhood graph. We're using the PCs because remember, the PCs capture all or most of the variance in the data. So we want to use those to compute our neighborhood graph because it's just going to be more, what's the word? More easy to interpret. The neighborhood graph is quite literally in the background. The algorithm is lining cells up in multidimensional space and saying, okay, this cell is similar to this cell based on its transcriptomic profile, which is represented by a particular PC 1 through 5 or whatever. And then we are going to visualize this graph, actually. Importantly, we're going to do a UMAP graph because that's a two-dimensional representation. So this is happening in some weird multi-dimensional space. And then this output, we use UMAP to project it into a two-dimensional space, which is what we can interpret as humans. So run that. There's a lot going on, so it takes a little bit. Okay. So this is what our data looks like. And we can see, so, oh yeah. So importantly, I decided to color mine by disease. I mean, you can do whatever you want. You could probably do cell type and see what that looks like. This is off script. I have not done that. Okay. So that's what that looks like. And what you decide to color by essentially just depends on the biological question that you're asking. If you're interested in disease states, then you color by disease states. If you're interested in cell types, quantifying gene expression across different cell types, then you color by cell types. The world is your oyster. I'm going to switch this back to disease because that's what we're talking about here. Okay. So we see our 3 disease states that we knew beforehand were represented in the data. Blue corresponds to cells that were derived from someone with Alzheimer's or people actually with Alzheimer's. This yellow color, orange, corresponds to those cells that were derived from folks who have breast cancer. And then the green corresponds to our healthy or normal cohort. And at this point is when I would try to be interactive again and say, huh, well, how would you interpret this UMAP? Um, I'm just gonna answer. And I will say that so when you do a UMAP, depending on what you decide to color by, you should see very distinct clusters. There shouldn't be overlap unless, well, I guess it depends. The answer to a lot of things in biology is that it depends. But for our purposes here, the fact that we're culling by disease, because we know that there are three distinct disease states in our data, we should see three distinct clusters. And I think that holds true. We've got breast cancer up here. We've got our normal folks sort of dispersed, but they're still pretty discrete until you get to this point and you see that this entire cell population, the Alzheimer's cell population, is covering this section of normal cells here. So what that means is that the transcriptomic profile of folks with Alzheimer's overlaps with folks who are healthy. In plainer terms, it means that the same genes that are turned on in people who have Alzheimer's are turned on in people who are healthy. Which, if you are all still here with me, is kind of odd. Because why would you expect that the same gene programs are turned on in healthy people, presumably me and you, and folks who have Alzheimer's? Like, how do you reconcile that? Like, are we treating the right things? What type of targets are our drugs geared towards for people who have Alzheimer's? These are all valid questions, which is why I decided to do a little bit more digging. This was not planned, by the way. I just did this and happened to see this overlapping cluster. I decided to do a little bit more digging and essentially create this table, which shows cell types by diseases. And one thing should stand out to you here, because it did for me. If you look in this row of mature microglial cells, this is the only cell type represented in our Alzheimer's population. These cells are not represented in our breast cancer population at all. And some of them are represented in the normal population. So if we go back up here, it's pretty obvious to us now that there's overlap in this particular region of our UMAP because these are all microglial cells that are present in both Alzheimer's, folks with Alzheimer's, and folks who are healthy and normal. And that makes total sense. But this is a prime example of, yes, you want to do a traditional single cell analysis pipeline, you want to do all the commonly used methods, but make sure your data makes sense. Definitely don't neglect the biology because that would be, if we had overlooked this, there are many hypotheses we could have made just based on this overlap, not knowing that it's actually just the population of microglial cells, which are very much present in healthy human beings. Okay, how am I on time? 15 minutes left. Mm, I'm probably going to run through this last bit a little more quickly than I had anticipated because I want to try to leave. I see the chat is sort of going off. I just can't see the messages. So I want to leave maybe 5-10 minutes towards the end for questions. But this is something else that you can do as part of a single cell analysis pipeline, which is again pretty standard, is you can use this function rank genes groups as part of the ScanPy package to do a differential gene expression analysis. And this just does a t-test in the background and compares two groups and assigns what is it, a t-score or a z-score to that interaction. So here on the left-hand side, we're looking at genes that are upregulated in folks with Alzheimer's because our normal is our reference. So whatever we see is going to be relative to our healthy individuals. And then on the right-hand side, we're looking at genes that were found to be upregulated in folks with breast cancer and not in healthy individuals. This is really powerful for, I think I mentioned this earlier, but it's really powerful for doing like drug treatment experiments, comparing group A to group B. Honestly, in any situation where you want to compare one group to another group, a differential expression analysis is powerful because it's just a t-test and some plotting to make it look pretty. Okay, so real quick, maybe let's just shift from thinking about individual genes to signaling pathways. So this is like the next level of your analysis. Up until this point, we've been talking about individual genes, maybe even individual cells, but sometimes those genes are part of regulatory signaling pathways that are important. And maybe if you're in the drug development space, then that particular gene, yes, it's important, but perhaps some of the upstream or downstream genes in that signaling pathway could also be important, could also have therapeutic benefit. So let's do that real quick. This is the exact same rank genes groups function as this. It just has this data frame decorator, so it turns it into a data frame, which is what we need. A lot of these functions they take very specific inputs, and so you need to keep that in mind as well. So this gives us data frames. Just want to show you what that looks like. So now we've got genes and they all have associated t-scores, as well as a log fold change. I don't know why that's a nan actually, but they also have significance values, which just at first glance do not look great, but that's okay. We knew we weren't deriving any biologically meaningful insights from this data. So now what we're going to do is feed both of those data frames that we created up here, one for Alzheimer's and one for breast cancer, to this pre-rank function from the gene set enrichment analysis Python package. And what that does is it takes the gene names and the log fold changes and it sort of maps them against different gene sets. These are publicly available gene sets. In particular, we're just using this one, the Go biological process from 2023. This is like a generalized gene set. There are disease-specific ones, treatment-specific ones, that sort of thing. And what this does is it goes through the list of genes that we give it from our data, and it maps them to these predetermined gene sets. And if our gene is in a gene set, then it assigns it an enrichment score, a high enrichment score. If it's not in the gene set, it assigns it a low enrichment score. So this is the output of the pre-rank up here. We can plot this, but none of these were significant. And the plotting, you need to meet a minimum significance threshold, so I didn't plot them. But we can look at the tables. And what those look like is that you've got these terms which correspond to the pathways or the gene sets that we referenced up here. And then they've all got an enrichment score, a normalized enrichment score. For Alzheimer's in particular, if it's a positive score, then it's upregulated in Alzheimer's. If it's negative, it's upregulated in normal because our normal is our point of reference. And then importantly, it also gives you sort of a list of the genes that it determined to be part of these particular pathways. So this is what that looks like for our Alzheimer's samples. And this is what that looks like for our um, breast cancer samples. So again, importantly, none of this is biologically relevant. These p-values are abysmal. So are these FDR values. But hopefully that's the end. Hopefully you guys have a better understanding of a single cell analysis pipeline, an end-to-end single cell analysis pipeline. Hopefully you know the value of doing things like QC, quality control, why transformations like normalization and log transformation and scaling and dimensionality reduction are not only important but integral when doing single cell analysis. Hopefully you think a little bit more critically about the code that you're running and how it relates to biology. And hopefully this was helpful. Um, that's it. I'm gonna stop the share.
Thank you, Anissa. While you're stopping the share, we have about 8 minutes left for questions. Um, I know there were some questions in the chat, but I want to let folks come off mute and ask their questions directly to Anissa so we can have some conversation. If you have questions, go ahead and just raise your hand in the on the Zoom so we can call on you. Go ahead, Fatima or Fatima.
Yes, do you hear me?
Yes, go ahead.
Okay, thank you so much. It's really helpful. My question is like in the pathway analysis, uh, uh, now the pathway that show up, they never reach statistical significance. What if I'm interested in a specific gene within distributed all over these analysis, these non-significant pathways? How relevant if I just like test the compare, for example, different treatment across all using only these to find differences between treatment? Can I do that? Like single genes, for example, or a couple of genes that representing only one protein, for example. Can I do that?
So let me just say that back to you. You're asking if you can do a pathway analysis on single genes instead of a data set of thousands of genes?
Um, Uh, okay, so I'm gonna say it in a different way. So, for example,
Okay.
let's take one pathway. Let's say, for example, innate immune, like interferon gamma pathway. Okay, um, there's a lot of genes representing this pathway. This pathway come up as negatively as non-significant, but I'm
Mhm.
interested in a specific set of gene that representing one protein within that huge pathway. Let's say
Mhm.
that. Can I just like look at those to see if there is significance um, between my treatment only for that protein, not the whole pathway? Does that make sense?
I think so. I mean, it sounds like you'd probably have to do that manual. Or are you asking if you can isolate a single gene that encodes a particular protein from a pathway enrichment analysis and just see if that one gene or two or handful of genes is significant?
Yeah, is that relevant to do that, or like I mean, stat scientifically relevant to do that, or and I think I can do it simply, right? I just like make sure like this protein is presented by these genes, and then I
Right.
do differential expression, right?
Right, right. So computationally, yeah, you can just extract a single gene of interest or a handful of genes of interest and evaluate whether those are significant or not. If it scientifically, if it makes sense, honestly, my answer to a lot of science is it depends. I'd say it depends on one, it depends on your sample size.
Uh-huh.
Because if like we only did this analysis on 5,000 cells. I mean, even if you get a significant hit from a data set that only has 5,000 cells, it's really not that reliable. Regardless of whether you're interested in just one gene or a handful of genes, just overall, the data set size or the sample size is going to be really important because you want to make sure your experiment has enough power. So like you
Yeah.
could have a gene, you could identify a gene that is statistically significant in a pathway of interest, but if you're only analyzing like 10,000 genes, I don't think I wouldn't pursue that gene, if that makes sense.
Okay. So probably I need to have replicate, biological replicate and other like a lot of cells.
And
Okay.
technical replicates,
And
all the
technical
replicates.
replace. Yeah, yeah. Oh, thank it sounds good. Thank you.
Hmm.
Thank you. And we have another question in the chat. What has been the most interesting work you've done with computational bioinformatics?
Oh, hmm. I think my grad school work was pretty cool. So there's a tool out there called Cell Chat that uses single cell data actually to infer communication between cells. So you can feed the algorithm, I think it's a binary classifier with something else, but you can feed the algorithm single cell data, and the output is that, okay, oh, thanks, Carmen. The output is it tells you, okay, well, these T cells are actually talking to these B cells, which are actually talking to these, I don't know, whatever other cells. So that's helpful for doing things like trajectory analysis. It's big in like the developmental bio stage when people are trying to figure out what gene regulatory programs control different cell differentiation stages. So like how do you get from one cell, an embryo, to the multicellular organisms that we are. So that was cool at the time. I think that came out in like 2021 though. So
Awesome. Thank you. Both the next two questions both have about AI in them. In your opinion, with the prevalence of AI tools, do you think that they will affect bioinformatics jobs negatively? And or how do you think AI will affect this field?
yeah, that's good. Very timely. Um, the short of it is that AI is already impacting bioinformatics. I mean, I mentioned really briefly, I don't know if you guys caught it, that I use Cursor sometimes. I showed you VS Code today, but sometimes I use Cursor, which is an AI assisted code editor. And I gotta tell you, it's made entry-level bioinformatics roles pretty much non-existent. Like you're expected to come in with a certain level of proficiency now, which I don't think is a good thing because it's now the barrier to entry bioinformatics, biotech is super high, unnecessarily so. So yeah, but I will say it's extremely helpful for like bioinformatics professionals because it's definitely helped my productivity and my output. You just need to be like really vigilant with using those tools because smart tools doesn't mean stop thinking critically about things.
Yeah, thank you. And we have time for one last question. I think with the folks who are on this call, I think it's really important for them to know and understand how did you get into this, and um,
Mm.
did you know that this was for you? And once you got into it, how did you keep going?
And what type of educational background do you need to break into this field?
Yes.
Hmm. Well, I got in because I quit my PhD. My PhD was in computational genomics, and I quit my PhD in 2022. Getting into biotech, I'm in biotech, but getting into biotech, biopharma, R&D, all of the things in 2022, or just data science and AI in general, was much easier than it is now. I think if I were just graduating, if I had stayed and I was graduating with my PhD now, like a lot of my friends who I left in the program, you gotta differentiate yourself. Like it doesn't matter if you have a PhD in immunology or a PhD in computational genomics because you're now being pit against this mid-level professional who's been in the space for I don't know, 5 to 10 years, plus AI. So now that's really like it's, I don't know, the landscape has changed so much. But at a base level, I would say you need the biology. The biology is arguably more important than the code, especially with AI right now. So you need biology domain expertise. I would recommend computer science expertise. So if you're at undergrad level, double major, biology, computer science. If you are sort of out of school and you're a professional and maybe you have a degree in biology, then you can definitely teach yourself to code. There are too many online resources now, so there's really no excuse. So you can upskill if you have the biology. I think it's harder if you don't have the biology. Say you have a computer science background and now you're trying to break into biotech. It's not impossible, but having a GitHub portfolio is now not optional because hiring managers need to be assured that you can do the work regardless of your background. Hopefully that's helpful.
I think so. Um, Amazing. Thank you so much. As you all can see, I put, as well as Carmen, put some information on how to connect with Aneesa, as well as with Melanin Science, as well as with Hack the Hood. So please make sure that you connect with all of us. One more thing that I would like to ask is if y'all can please, please, please take a moment and fill out our survey. You all will receive an email as well from us to fill out this survey as well, but please take a moment and click the link there. But we really appreciate you all for spending time with us this evening, morning, or wherever you are in your time zone. Please thank you to Aneesa and Carmen from Melanin Science, Aneesa and ImpaktBio for spending your time with us and putting on this amazing demo and this learning opportunity. Thank you all for attending this session with us and have an amazing rest of your day, night, wherever you are, um, and have a amazing rest of your day and evening. Have a good
Thank
I'm up until like 1 a.m., so whoever you are,
one.
thank you for being here. But it
Thanks,
was a pleasure.
folks.